revolutionizing the DNA isolation process. DNA defines everything that has ever lived. It is the cornerstone of who and what we are. DNA technology is also the driving force behind disease diagnosis, forensic science, and the identification of bacteria and viruses. But how is the DNA accessed from the cells? Current techniques to isolate DNA in preparation for amplification and analysis are complex and costly. Our next-generation solution is set to revolutionize this process. Current lab protocols used to isolate DNA. The current process begins with the breaking down of the cell membrane using a lysis buffer to release the DNA. To prevent any degradation of the very delicate DNA, magnetic or silica beads are typically added to bind the free DNA. These beads are collected and washed to remove any enzymatic material. The final step is to add a buffer to release the DNA from the bead. This leaves the isolated DNA, which is suitable for amplification and further processing. This 5-step process takes between 45 and 60 minutes to complete and requires costly equipment such as a heater, centrifuge and vortex, as well as highly trained staff. We believe that this process can and should be simpler. Introducing PCR Direct, ultra-fast DNA release from Arcis BioApps. PCR Direct is an innovative 2-step protocol designed to radically simplify the process of releasing DNA for any further processing, such as amplification. The process only requires a very small sample, just 30 microliters. And once a sample has been taken, it is simply added to the pre-prepared tube 1, which contains Arcis buffer. The tube is thoroughly mixed for 1 minute. To complete the process, 2.5 microliters are taken from tube 1 and added directly to tube 2. After thoroughly mixing for 1 more minute, the sample is ready to be added to a master mix for amplification or further analysis. By slashing the time and equipment required to produce samples, we have been able to significantly reduce the typical cost and complexity of isolating DNA. Better still, PCR Direct is a multi-function product that's applicable to all areas of the DNA testing market. It's suitable for releasing and amplifying DNA without further processing directly from fresh blood, frozen blood, plasma, cell cultures, buccal samples. PCR Direct is also proven to be effective in releasing DNA from cells and capsids, including bacteria, viruses, fungi. A fantastic opportunity. With a variety of market sectors demanding simpler and more cost-effective sample preparation, the potential for PCR Direct is significant and growing. PCR Direct has been developed to be used independently or incorporated into proprietary and third-party systems. It's already validated as a reagent for rapid cell lysis across cell types and fully compliant with all common DNA amplification methodologies. Compliant with endpoint PCR, real-time PCR, pyrosequencing, Sanger sequencing, cloning. What happens next? We recently launched a research-use-only ultra-fast DNA release reagent that's currently under evaluation by a number of academic and research institutions. Research-use-only products provide a stepping stone into the diagnostics market and a low regulatory path to accumulating more independent evidence. We are also looking for strategic partners for distribution and also to develop automated and point-of-care applications. All the signs indicate that this is a great time to bring a new and innovative product to market.